Protein structure. Now, it's important to note at this stage that this is a chemistry course. So many of you will have looked at these things in biology, and biology takes a particular approach to things that is slightly different to what we emphasise in chemistry. We're interested in bonds and functional groups and their interactions in chemistry much more so than would be covered in biology. So the content is overall the same, but different emphasis. So we look at primary, secondary, tertiary and quaternary structure, different levels of protein structure. First of all, the most basic is the primary structure, simply the sequence of amino acids in the protein chain. So again, from amino end down to carboxyl end, we just have that sequence of amino acids. This is what DNA codes for, and all the other properties basically are going to stem from this order. Now, the amino acids, of course, are joined together by covalent bonds, that peptide bond between each one, strong covalent bonds. If you want to break those bonds, if you want to break down the primary structure, you've either got to do so pretty ruggedly with acid and high temperatures in a test tube, or you've got to use specific enzymes like proteases to break them down biologically. But strong bonds, hard to break. Secondary structure are repeating patterns of structure, producing shapes, repeating shapes in the protein chain. Now they're caused because every peptide link contains a hydrogen bonding group. That NH on the secondary amine is a hydrogen bonding hydrogen. It can link up with a C double bond O, the oxygen there, from a nearby peptide bond and form a hydrogen bond. You get repeating patterns of these hydrogen bonds formed up and down the chain to get repeating patterns of protein structure. So the classic alpha helix of keratin found in hair, beta pleated sheet in silk, etc. So chemistry, we emphasise the hydrogen bonding between the C double bond O and the NH, the repeating patterns of those. Tertiary structure is the overall three-dimensional shape of the protein. Now these are really important, and they're determined by the myriad of interactions between different parts of the protein chain. So as a protein is being manufactured in the ribosome, it comes out of the ribosome and folds in a particular way. That's very important, that initial folding process, as it gives it its tertiary structure, gives it its form and function. Now all of the intermolecular forces combine to produce this structure and enable it to fulfill its function. So, depending on what the R group is, we can have lots of different things happening. We can have hydrophobic interactions, dispersion forces between two non-polar R groups. We can have hydrogen bonds formed, where we've got a hydrogen bonding proton in the polar groups like the amides and the hydroxyls. We can get hydrogen bonds formed. We can get ionic bonds formed, where we've got an acidic and a basic amino acid side chain. And we can get disulfide bridges formed when two cysteine molecules actually physically react together to form a covalent bond. We also get dipole interactions and iron dipole interactions as well. So let's go through them. Ionic interactions, where we've got a basic group in its cation form and an acidic group in its anion form. They're full on iron, so there's going to be quite a strong attraction happening there. Hydrogen bonds, where you've got the two hydroxyl and amino protons able to find a lone pair on things like the Citadel Bondo. Then, as I say, if you've got two cysteine amino acids, each of them have got an SH group, so they actually react together to form a proper full-on covalent bond. Polar side chains, of course, will interact via dipole bonding. And as I say, dispersion forces are still going to operate between R groups, non-polar R groups. So the disulfide link, two cysteine amino acids on adjacent parts of the chain. The two SHs react together, so you lose the two hydrogens and join together the two sulphurs. Disulfide bond, disulfide bridge, full covalent bond, giving a firm solid structure to the protein chain. Now that's tertiary structure, the overall shape of the amino acid. Quaternary structure is when you combine two or more polypeptide chains. So the functional unit is made up of a combination of different chains. Hemoglobin is an example of that, where four individual protein chains, four individual globin molecules interact with each other to form with Fe ions stuck in there, and they form the overall hemoglobin molecule. So, summary with the chemistry emphasis. Primary structure is sequence of amino acids held together by covalent bonds. The peptide bond. Secondary structure, repeating patterns produced by hydrogen bonding between NHs and COs in adjacent peptide links. Tertiary structure, the overall three-dimensional shape produced by the various ranges of interactions, including dipole interactions listed in this. And quaternary structure, when a protein is made up of several different polypeptide chains. And the bonding that holds the quaternary structure together, often dispersion forces, but other types of bonding also occur. Okay, do those questions and submit your answers. I'll give you the solutions.